Good afternoon, colleagues, teachers, learners. Welcome to this presentation of Key Points Paper 2. I hope that we can emphasize some important things today and that it will be a teaching tip for you to prepare for the trial exams coming next Tuesday on the 7th of September when you are writing question paper 2. So in this session, we're going to focus on paper 1 and then also the topics of DNA, the topic of genetics, and then the one of evolution. So without further ado, let's start then on the key points of paper 2, DNA Code of Life. I hope you can see my PowerPoint. And DNA Code of Life is 27 marks of paper 1. So we are going to look in this question paper 2 on the structure of DNA and RNA. Replication, transcription and translation, always very, very important in question paper 2. Not your own words as described in the exam guidelines. Sorry for that. This is very important to note that these three are always evaluated in question paper 2. And they are always evaluated in such a way that the allocation of marks is according to your exam guidelines. So something that I just want to emphasize when we come to DNA replication, we must talk about the DNA double helix. Not the helix, not the DNA double helix that unwinds. Another one is then unzip, and then to form two separate strands. We find that learners are leaving this mark out. They skip right from there to both strands serve as a template. And then the next one, both strands serve as a template to build a complementary DNA using free DNA molecules. You can see the DNA nucleotides. Remember now we are talking about DNA replication. So we cannot state in our answer using free nucleotides. It's DNA nucleotides because it's DNA replication. And this results in two identical DNA molecules. So this is very important because now what happens at the end of the year in the marking session, sometimes they decide unwinds and unzips allocated 1 mark. For both sentences stated, then they allocate 1 mark. Sometimes they decide this one and this one is also 1 mark. So learn the learners to write it in this sequence according to this wording, and they must state everything. Although it counts 6 marks, then they decide at national sometimes what do we allocate and what not. But this is the way it is stated in the exam guidelines, and that the learners must also identify and discuss or describe or explain. So please, according to these notes, they must describe DNA replication. Every time it's either DNA replication, maybe transcription and translation, but they always evaluate these. Again, with transcription, as stated in the exam guidelines, we have a double helix unwind. The double helix DNA unwinds. Unzip, 1 strand is used as a template to form mRNA using free RNA nucleotides. Then again, and therefore a complementary to the DNA. Sometimes, and it's not wrong, learners wrote to form a mRNA or to form a complementary mRNA, and then for that one sentence they will allocate 2 marks. And at the end, the mRNA now has the coded message for protein synthesis. So in this sequence again, we need these keywords to be in the answer as stated again in the exam guidelines and not their own explanations. Translation, same one as translation. as I stated before, always either translation, either transcription in the exam paper 2. And again, the wording according to the exam guidelines must be each tRNA carries a specific amino acid. When the anticodon on the tRNA matches the codon on the mRNA, don't just write when the anticodon matches the codon. You must indicate that the anticodon is on the tRNA and the codon is on the mRNA. And then the tRNA brings the required amino acid to the ribosome, and then we have to say amino acids become attached to one another by peptide bonds and to form a required protein. Again, this is as it is in the exam guidelines. So either these replication or translation or transcription every time evaluated. Then there's the normal questions that they always ask on replication. We know that this is a DNA molecule because it is double helix, or in this case it have thymine. So they must be able to identify it as a DNA molecule. They have to identify that this one is allocated in the nucleus. DNA is also allocated in mitochondriums, and we have also DNA that is in plant cells that is allocated in the chloroplast. You will see in the new exam guidelines they especially refer to DNA in the chloroplast. So maybe the question will be, where do we find this strand in plant cells? And then they must allocate it as chloroplasts. Learners have to identify the T is for thymine and the G is for guanine. Identify X, that is the phosphate molecule. Remember, the sugar molecule is allocated next to the nitrogen base, and A is the sugar molecule, that is deoxyribosis. Not just sugar molecule, but deoxyribosis. So a straightforward one in DNA to identify this DNA molecule. But look at this question. How many nucleotides are represented in this diagram? Now the learner must identify that this is a nucleotide because of the phosphate and the sugar molecule and the nitrogen base. And now there's 1, 2, 3, 4 nucleotides in this one. So a level B type of question, because you must know what is the nitrogen base nucleotide and what is the composed of that, phosphate, sugar and the nitrogen base. And now we can count 1, 2, 3, 4 nucleotides. All right, and then also write down the nitrogen bases from top to bottom as indicated by the arrow of the complementary DNA strand of this molecule. So T, A, G, C, A, T, C, G. So type of questions that learners can expect in this question. So I just want to emphasize something on 1.5.3. They ask that you must allocate the complementary DNA bases from as the arrow indicated. So the learner can either write it down like the arrow or in this case. But please emphasize on the learners in marking session. That is the question. Give the answer. Don't repeat the question. Don't go and write T, G, A, C, then an arrow, and then your answer. Or what else? Because remember now we are actually allowed to mark the first answer. So many times they write this T, G, A, C with an arrow gives you A, C, T, G. And then we have a huge discussion every time at marking centums to see, we ignore then the principle of mark the first answer in this case. So it's a very narrow line to walk on. So I'm just afraid that the next time at DBE they say no, we mark the first answer. But in this case we can clearly see that the learner is indicated that is what stands in the question and that is my answer, but they must clearly indicate that for us. The next one, same type, just to emphasize to you what is a nucleotide, and they have to identify it, give the names of the following parts, a typical question that they can ask, and then again, name 2 organelles in a non-dividing human cell where the nucleic acid mentioned is found. Remember now the nucleus and the mitochondrium, and in plant cells, the chloroplast. Okay. Again, typical question, DNA replication, mention what is represented by the diagram. We know it's DNA because it's helix. Give one reason why this process is important. The DNA replication, the importance of DNA replication, always very important question that learners must be make or must be or may explain this in an exam paper. So please look at that one also. But this is typical questions on how they can ask DNA and DNA replication. All right, so again, give one reason for this process is important. To emphasize on this one is I find many times when I'm marking that learners get confused on the importance of replication and the importance of meiosis. They get confused with those two. So stipulate the importance of DNA replication and then the importance of meiosis, the process of meiosis. So again, there's the table. Just to indicate that we can ask the differences between DNA and RNA. DNA and RNA is also very favorable question to ask, the role of DNA and RNA. Not the role, the difference between DNA and RNA. This simple diagram to illustrate protein synthesis, it is very important that learners must be able to identify these structures. So this is DNA, the replication of DNA. This molecule is a messenger RNA because it's a single strand. This one, in this case, we have a lot of amino acids. So this is a peptide bonding or peptide chain. That one, that is alone, that one there is an amino acid. This structure is called the tRNA, this one. This one is the bRNA because it's a single strand. Also, we must indicate in an illustration like that this is the ribosome where protein synthesis takes place. And then again, we must indicate that this is transcription, and the one that's taking place at the ribosome is translation. So always remember to take a lot of different illustrations so that learners can identify the DNA molecule, etc. So in this case, also important, then they can ask, explain or tabulate the differences between the DNA and the bRNA, or they can say the difference between A and B, or they can ask also a question in this to explain what is the role DNA plays in this specific protein synthesis. So this is protein synthesis where we have transcription and translation, but what is the role of the DNA molecule in this process? Now remember, bRNA gets the coded message from DNA to form a specific protein. So DNA gives the coded message. The other function of DNA is they play a role in DNA replication, but in protein synthesis, DNA plays the role where it provides the coded message for messenger RNA to form the specific protein, to form the specific protein. So this, very important, always evaluated in question paper 2, illustration of protein synthesis, and learners have to identify the different parts. Again, there's one. So DNA, bRNA, which is a single strand, tRNA, X allocates a tRNA, the ribosome, this process is transcription, and this process is translation. Also indicating that this 3 is the codon because it's on bRNA, and those 3 will be the anticodon because it's on tRNA. Again, a question, tabulate 2 differences between DNA and RNA. So there's a whole lot of differences between a DNA and the RNA. Be sensitive, be very sensitive if the question stated that you only must describe the differences in the nucleotides of a DNA and the nucleotides of a RNA. Then we're only going to refer to the sugar, and we're only going to refer to the nitrogen base, because that is what the nucleotide consists of. Nitrogen base and the sugar. Then the sugar is deoxyribose and the sugar is riboses. And then we have the difference because this one is thymine and this one is uracil. So be sensitive on that one. And again, also, what role does DNA plays in transcription and translation. And then also a difference, I think it was one of the tests this year, one of the formal tests, or it was in a exam. But the other question also asked is, what is the differences between DNA replication and transcription? So in DNA replication, both strands act as a template, and in transcription, only one strand acts as a template. And then there's the difference on the adenine that binds with thymine, but with transcription, it's adenine that binds with uracil. So just be sensitive for that one also. So I just want to summarize something here. The role of DNA replication or the importance of meiosis. They must be able to separate these two from one another. The differences between DNA and RNA. Also the differences between DNA replication and transcription. And then the role DNA plays in the whole process of protein synthesis. So that is the important things on DNA and RNA for 27 marks. Right, and then another one that I want to emphasize is the human karyotype. Either in a section A multiple choice question, or in section B, but the human karyotype is also always there. Either they will ask what is the autosome or what is the gonosome, or in this case you must indicate which is the autosomes and which is the gonosome. But a very important part of question paper 2 is the human karyotype. They can give you the diagram, and then you must identify that this is a karyotype. Or they can state that this is a karyotype, and then you must describe what is the term karyotype. And in this case, it's the arrangement of the chromosomes according to their number and their function. So karyotype, the definition, or to identify that this is a karyotype is very important. Then we have this one that is indicating for us that this is a homologous chromosome pair, one from the mother and one from the father. Then you also have to identify the autosomes, that is chromosome pair 1 to 22, and the gonosome is this one, pair 23, which can be XX or XY. So on this one, I just want to emphasize something. Now this is the karyotype of a male because it has an X and a Y chromosome. Let's stick to that. What is this karyotype? That is a male. The reason for that is it has an X chromosome and a Y chromosome. That is what I can see. Learners will write this is a male because it does not have an X and an X. So let's stay with what you see you must explain, not what you not see. So in this case, this is a male because it is an X in an X or X and a Y chromosome. So don't go there and explaining how a female looks like if they ask you, is this a male or a female? So in this case, it's a male. And then always chromosome pair 21, we have an extra chromosome pair on that one. That is trisomy or Down syndrome. So we're looking for 21 and for 23 in this karyotype, and they can ask you questions on this one. Another questions when they give you the karyotype is to ask you how many chromosomes are there present in this cell. So in this case there are 47 because there's an extra one. Or they can ask you how many is in a normal person, and then it's 46. And how many will be present in a normal gamete. In this case, this is a somatic cell. So in a gamete, then there will be in a normal gamete, there will be 23. In one of these gametes, one will have 25, or the other one will have 22. So be alert on that questions that can be asked on karyotype. A lot of different questions on this one. So in one of the other exam papers, I pick up, they asked the genetic makeup of this person. So the genetic makeup of a normal person is 44 plus XY. So in this case, it will be 45 plus XY. I'm going to say that again. What is the genetic makeup of this person? So the genetic makeup will be 45 chromosomes up till 22. It's 45 because there's an extra one plus XA. So that is also a question. And in this case, they can also ask how many autosomes are there. Up till there is 45 autosomes. How many gonosomes are there? X, Y, there's only 2 gonosomes. So be aware that the learners must note what is autosomes, what is gonosomes. Autosomes are up to 22. And how many chromosomes are there? 45. This gonosome have only 2. So that is a mouthful on karyotype, but it can count up to 6 to 9 marks. It's actually very easy if they understand it and they know the difference between the autosomes and the gonosomes. They will be able to answer the question. Typical question. We have 21, the disorder is Down syndrome. This one is a male because it has an X and a Y chromosome. Identify the type of chromosomes, number 1 to 22, and as before we see that it is autosomes. All right. So in this case it's a male because it has an X and a Y chromosome. If we go to meiosis, meiosis is now fully part of question paper 2. It counts 21 marks in question paper 2. And what is the usual that we ask? The usual is they must identify the different phases, prophase, metaphase, anaphase, telophase, and they must indicate in which phase it is, 1 or 2. I just want to emphasize something here on prophase and metaphase, as meiosis play a very important role in variation, and also variation plays a very important role in natural selection. So if we look at variation, the role meiosis play in variation can be asked as a describe or explain question. Explain how meiosis can contribute to variation. Now remember, we don't have the long question at the end of the year anymore in section C, but they can break it up into smaller questions where you have to explain something. So when we explain meiosis, the role of meiosis in variation, we must indicate prophase 1 is where crossing over takes place, at the chiasmata, where genetic material is exchanged, resulting in 2 different chromosomes. I'm going to repeat that one. So prophase 1, crossing over at the chiasmata, genetic material is exchanged, and the result is 2 different chromosomes. So that is 4 or 6 marks. And then at metaphase 1, we have random arrangements of homologous chromosomes that results in different gametes. This one, prophase 1, results in different chromosomes, but metaphase 1 results in different gametes. So the role of meiosis, the role that meiosis plays in prophase 1 and metaphase 1 is very, very important. And I think that is a nice question to ask and in a question paper, and to be evaluated to test if learners know what is meiosis, the role of meiosis in prophase 1 and metaphase 1. So I want to emphasize that one. That can be asked in paper 2 because we have meiosis in paper 2, we have genetic variation, and we have evolution in paper 2. So meiosis. Typical questions in meiosis is to identify different parts of the process. This is anaphase 1, metaphase 1, and I just want to emphasize the difference here to you. In this case, there are no spindle fibers. No spindle fibers, so that is anaphase 1. And in this case, there is still spindle fibers between them, so that is metaphase 1. And then the usual questions to give the phases, also to give the labels. So we have anaphase, this is anaphase 1 actually, not 2. Metaphase 1, not 2. And then we have anaphase 2 here. We have anaphase 2 here. So in this case, this is a chromatid, this is a centromere, that is homologous chromosomes, and a centriole. The typical questions, crossing over taking place there. Again, typical questions, identify that it is metaphase 1, centriole, homologous chromosomes, spindle fibers. All right, another question that I will also pick up at some of the question papers, how many chromosomes are there? 1, 2, 3, 4. How many chromatids? 1, 2, 3, 4, 5, 6, 7, 8. How many centromeres? 1, 2, 3, 4. How many centrioles? 1 and 2. So that is another type of question that I can ask. In this one, how many chromosomes will there be at the end of meiosis? 4. No, 2. 2 chromosomes will be at the end of meiosis. How many cells? Sometimes they ask how many cells are there at the end of meiosis, and that gives us 4. So in this question, how many chromosomes? There will be 2, but be very sensitive. They ask how many cells will there be at the end of this meiosis process, then it's 4. So very careful reading when you answer question paper 2 on meiosis. Then again, identify the different phases, explaining how many chromosomes and how many chromatids, typical question that you are going to be evaluated, and to answer that. And then in this case, the importance of meiosis. Remember now, it's different from the importance of DNA replication, and this is straight from the exam guidelines, so they can learn it in the exam guidelines. And then another one, in the 2021 new exam guidelines, they stipulated it very clearly there, and the mechanism to introduce genetic variation is through crossing over and random arrangement, and then maybe you have to explain it. So I have this gut feeling for the end of the year, maybe they will ask this. I picked this question up in 2015 November exam, was it last evaluated, where learners have to explain meiosis as a source of variation, and they have to explain crossing over and random arrangement there. So be sensitive for that, maybe to describe or to explain it. All right, so on meiosis I have this last one. The other way they can ask meiosis is again in this sketch. They ask, they give us this, and what process is taking place? Crossing over. Where does it come? When does it take place? During prophase 1. Where does it take place? In the nucleus. But now also very important in this one is they can ask you, or the learners, they can ask the learners to draw a sketch or a diagram of a chromosome at the end of this process. After this process have taken place, you have to draw one of the chromosomes. So remember now, everything when you draw in paper 2 and 1, draw something, a graph or a table or a diagram, it must have a caption. So I can label it with a caption, and then you have to draw this picture of a chromosome. Then if you draw this chromosome, this leg will still be like this one, and then if you draw this leg, that T will be the same. What happens now, learners will write their capital letter T, but actually there's also a small letter. Crossing over did take place. They exchange genetic material, so the T stays the T. And then we look at this one. Then the G will exchange, the small letter G and the capital letter G will exchange. So this one will be a G. And then you have to draw the correct chromosome. Sometimes they will be coloring it in. For example, this part is colored in. And then when this two split again, this part will be the one that is colored in. Very important here is to draw the diagram as it looks on the exam paper, and the color, if it have to be colored in, the same amount of coloring in. If that one is only the tip was colored in, only this tip will be colored in, not the whole one. So just be sensitive on that one, and remember they must add labels to this again. So if they draw it and they color it in or exchange the genetic material, they will indicate the chromatid, a centromere, and this is the chromosome. So that is where the marks will be allocated. So in a different way, they can ask meiosis like this, giving homologous chromosomes where they are crossing over, as an example in this specific question. Another part of meiosis that is very important that you can identify or able to identify is this process that takes place here, and that is non-disjunction. Non-disjunction. So where the chromosome fail to separate. The chromosome, there's the chromosome and there's the chromosome, they fail to separate because there are two homologous chromosomes, and now we call it non-disjunction. One of the chromosomes did not separate during anaphase 1, resulting in gametes with one extra or one less chromosomes. So that is very, very important to look at and to be able to identify non-disjunction. Again, this one, also non-disjunction, that takes place here. And that is the failure of a chromosome pair to separate during anaphase 1 or anaphase 2. And that causes Down syndrome. Right. Then, on genetics, genetics can be very difficult because the learner must understand genetics before we can answer any of the genetic questions. So let's look at some of the very important keywords in genetics. So in this case, this is the basics, and we have to learn the definitions of these. If we don't know the difference between these definitions, then we won't be able to answer the question on genetics. So there's a lot of explanations and definitions, and maybe in textbooks there are a lot of definitions of this. But let us just look at genes. That is a small portion of a DNA molecule. Alleles is different forms, so you're looking for different forms. Genotype is the genetic makeup. Phenotype is the physical appearance. We must be able to identify that. The dominant allele, I will say something on dominant allele further on this presentation. We must identify the recessive allele, and what is the recessive allele? Heterozygous, homozygous, that is all part that we must identify. So if we look at a few of these definitions that I always ask on genetics, or one word biological term, the position of a gene on a chromosome is called the locus or the loci. The different forms of a gene is called the allele. The full set of genes is the genome, also a favorable question that asks. Different forms again, it's allele. The physical appearance of an organism is its phenotype, physical phenotype. The number, shape and arrangements of all the chromosomes is called the karyotype. So just look at the difference between the full set of genes is a genome. Full set of chromosomes are arranged according to their number and shape. That is the karyotype. So there's a difference. And then the appearance of an organism. The appearance of the organism is its phenotype. And as a result of its genetic makeup is genotype. So if this was left out, they only asked an organism's genetic makeup, it would be genotype. But now they ask the appearance of its genetic makeup, that is a phenotype. A segment or a part of a chromosome that codes for a specific protein, part of the chromosome is the DNA. Part of the chromosome is the DNA. A pair of identical chromosomes found in a diploid cell is homologous chromosomes. And then If you have two identical alleles, it's homozygous. If it's not identical, it's heterozygous. So this is the most popular questions that they ask and give the biological terms when it comes to genetic. Make sure that you are able to identify this and can give the correct biological term when you ask. DNA profiling, I really want to emphasize something here on DNA profiling. And the first one I want to emphasize is about when we identify the father of a baby. So, DNA profiling, usually they can give us the profile diagram like this, which is DNA profiling. Right. Now they can ask us, how do we determine the male, or what? You must determine the male. So look what we are going to do to determine the male. But I'm also want to alert you or be sensitive on the following. Maybe they will not give us the DNA profile. They will not give us this, but they will ask us, explain or describe how would you determine the father of a baby without giving the DNA profile. Then we look at the DNA bands of the baby. We look at the DNA bands of the baby. It's not we look at the DNA of the baby. It's not we look at the DNA profiling of the baby. We look at the DNA bands of the baby. And be sensitive when you are writing your exams. Remember now there's a time limit, you are stressed, and then we usually write only the DNA of the baby, and that is totally wrong. Look at the DNA bands of the baby. Identify all the DNA bands that are common with that of the mother. So we're going to look that one, and that one is common to the mother, that one and that one. This one must then be compared to the males. So at the last one, if all the remaining DNA bands of the baby correspond with that of one of the males, then that male is the father. Then that male is the father. So we're going to look at that one. It doesn't correspond with the mother, so it must correspond with one of the males. Also this one, it must correspond with one of the males. Same with this one, must correspond with one of the males. Now in this case, we can see that it is male 2. And now's the answer very important in this case, where we are going to say that the remaining bands, DNA bands, the remaining DNA bands of the baby that is not corresponding with the mother's DNA bands, all of them correspond with the male 2 DNA bands. So the baby, all the baby bands, all the baby DNA bands must be either from the mother or either from the father. So either you have to do it, or either you have to explain it like this. And I'm also thinking maybe they can ask this as a question, an explaining question. How do you use DNA profiling to do this? Determining which one is the male. So in this next slide, I want to show you what we did. We take the mother and the child and we eliminate the bands. So we eliminate those bands, DNA bands. Now we take the child's, the remaining bands of the child, and we compare that with the male. So in this case, this one and this one and this one and this one compares with the male, and this one also compares with male 3. So male 3 is the father. But look at the allocation of marks. How many marks are allocated? So what happens now, learners wrote male 3 is the father, right? And now they have to explain it. And now they say the DNA of male 3 and the DNA of the child correspond. Totally wrong. Totally wrong, right? So we must indicate that the remaining DNA bands of the child that it does not correspond with the DNA bands of the mother. All of them correspond with the DNA bands of male 3. All of them, all of the remaining bands, DNA bands of the child, correspond with the DNA bands of the male 3. I hope that is clear, because that is also a really technical thing in the end of the year paper. So I just want to emphasize this again to you. So this was asked in November 2020. So who is the criminal? The possible criminal is Jenny. All right? But now explain your answer. Why is Jenny the possible criminal? So this is a DNA sample, and if you look quickly, you will see that all Jenny's DNA bands correspond with the DNA sample found on the crime scene. All right, so now the explanation is very important for me. So we start with to say, in our explanation, we say all Jenny's DNA bands, not all Jenny's DNA, not all Jenny's bands, it's all the DNA bands of Jenny correspond with all the DNA bands of the sample. Not Jenny's DNA is the same as the DNA sample. Not Jenny's band is the same as the bands of the DNA sample. All Jenny's DNA bands corresponds with all the DNA bands of the DNA sample. So we must refer to the DNA bands of the DNA sample. We must refer to the DNA bands of Jenny. And then the last one is state one other use of this technique and prove paternity, tracing missing persons, identifying of genetic disorders, establish family relations, matching tissues for organ transplant. It's not only we use it for organ transplant. Sometimes the learners wrote very cryptic notes, organ transplant, dead persons. So full sentence, yeah. Not just paternity, proof of paternity, tracing missing persons, identification of a genetic organ, not just genetic disorder. What about genetic disorder? So let us write full sentence here, because that is also a very technical point where the learner only write paternity, and but the memo says we need to have proof of paternity. So again, I just want to emphasize this again, really, people, because this is where our learners and our top candidates, our level 7, level 6, 7 learners lose marks. They know what to do. They know Jenny is the criminal, but the explanation is that Jenny's DNA bands, all of them, correspond with all of the DNA bands of the DNA sample. I think it was on a broken window. There is it. This DNA sample of the broken window was there. So please look at that wording I'm using. DNA bands of Jenny correspond, all of them, correspond with all of the DNA bands of the DNA sample. So, okay, again, a lot of information just on this, but sometimes it counts 6 marks or 3 or 4 marks, and then the learners don't allocate all the marks because of that technical sentence of DNA bands. So enough on that one. The genetic cross, quickly just to show you where we allocate the marks. P1 is on top and F1 is on the bottom. The P1 must have the phenotype, it must have the genotype. The F1 again, it must have the genotype, it must have the phenotype. And I'm saying this irrespectively of what they ask. Sometimes they ask a ratio, or sometimes they ask a percentage or whatever. Before we come to the answer, we must write it like that. Phenotype, genotype. Then meiosis and fertilization, not fusion, not fusion. Fertilization, meiosis, between meiosis and fertilization is the gametes, and we must indicate the gametes. The phenotype is tongue roller and non-tongue roller. Please let them write out the whole explanation or the whole phenotype that is asked. A male, if it is a genetic cross with a sex-linked disorder, they must indicate male or female also. They don't have to use this, I call it spider web method. They can use a Punnett diagram also, but that is where we allocate the marks. And then at the end, when we ask maybe what is the percentage or what is the ratio, let the learners write the ratio underneath, here underneath, and underline the answer of this question. Because sometimes they forget to answer. They do the cross, but they forget to give the answer at the bottom. So compulsory marks, meiosis, and phenotype, and F1, even if that is everything is wrong, at least 2 out of 6 they will receive if they allocate it in the right positions. So that is just that one. So this is a simple example, but just to show you, they can use also this type of crossing, meiosis, fertilization, meiosis first, then fertilization. All right. Hybrid genetic cross is also very important. There's a lot of examples that your learners can do. So in this case, I just want to emphasize that we are evaluated 2 genes. It's the color of the fruit and then the height of the fruit. In this case, there's 2 genes, but there's 1, 2, 3, 4 alleles. There's 2 genes, but there are 1, 2, 3, 4 alleles. So typical questions is to write down the genotype and write down the phenotype, write the possible genotypes of gametes of plant A. When we have gametes, we have to sort the gametes. Phenotype, ask the genotype. I want to emphasize something here. If you look at this question here, or statement in tomato plants, the allele for red fruit is this, dominant over the allele for yellow fruit, etc. Now, be sensitive on a question like this one. What is the recessive allele for fruit color? What is the recessive allele for fruit color? And then the learners wrote yellow, which is totally wrong. The allele is small letter R. What is the recessive allele? What is the recessive allele for fruit color? And the allele in this case is R. What is the dominant color for fruit color? And then it's red. What is the dominant color? Then it's red. But when I ask allele, then they must allocate the single allele that is represented. So there's a lot of examples. Again, example, typical questions, give the phenotype, give the genotype, that they must answer. So they must very carefully know which is dominant, which is recessive. Again, 2 genes, but 4 alleles that is evaluated. And they can do all these examples if they want to. Again, in this case, just to show you 2 genes, plant height and stem color, but 4 alleles are evaluated. And then they can give it like that also. And you must, the learner must be able to complete the table and that one, capital letter T, small letter t, capital letter B, small letter b, and that is the type of questions on dihybrid crossings, because we have 2 genes, that is a dihybrid crossing. So if you just can emphasize that also on the learners, we're always writing the capital letters first and we group them in this specific order, T and B, and in this case, that is the order of allocating the alleles, the different alleles. Very careful reading when they have a diagram like that, so that they can be able to identify the dominant and the recessive alleles, phenotypes, etc. There is again an example of that, where the learners must be able to answer some of the questions. Again, identify the phenotype, give the possible gametes. It's regular questions always that we always ask and must be evaluated. Again, another one, I think it's the same as the other one, previous one. And again, dihybrid crossings, another way to evaluate it or ask the questions on this one. You can see it's all, this is the same, the dominant phenotype for flower color, then we must name the color. If they ask the allele or the genotype, you must refer to the allele or the genotype. Then let's do dominance. There's 3 types of dominance. The complete dominant. Then I also want to emphasize this to go and look at the explanation in the exam guidelines. I know in June exam, I can't remember, is it incomplete of codominance that was asked. But look at the definition as stipulated in your exam guidelines. So this is complete dominance. One allele is dominant and the other one is recessive. So that is the explanation. If we look at codominance, both alleles of a gene are equally dominant, whereby both alleles is expressed in the phenotype. So this one is expressed, this one is also expressed in the heterozyte condition. So this one is a heterozygous organism. Both of them are equally dominant and both of them are expressed. Now what happens in the exam is the learners wrote both alleles are equally dominant and equally expressed in the phenotype. It's not equally expressed in the phenotype. They are both expressed in the phenotype, but it's not, sorry for that one, it's not necessarily equally expressed. So, but this is codominance. They are expressed in the phenotype, in the heterozygous condition, but not equally. So be very, very careful to write the equally. They are both equally expressed. They are not both equally expressed. They are expressed. And I will show you another picture later on, so we can differentiate between that one. And then the intermediate form, incomplete dominance, neither one is dominant over the other one. Neither one is dominant, resulting in an intermediate phenotype in the heterozygous condition. That one is added in the heterozygous condition. So in the previous examination guidelines, this wasn't stipulated like this. So an intermediate form, purple, pink, red and white. So there is an intermediate phenotype, also in the heterozygous condition. So just to show you, they can ask it also in a monohybrid crossing. Then you have to remember that irrespectively of it is codominance or incomplete dominance, we use both letters as capital letters. So this, again, just where we allocate the marks, but this one I just put in to show you that they can ask it in a crossing, but remember then it's all, we use capital letters. We use the capital letters to show the dominance. Incomplete dominance, intermediate form in the heterozygous condition, and we have both alleles are dominant, and both of them are expressed in the phenotype. Not equally expressed, but both of them are expressed. So black is black, and white, there is white, and there is the white colors in codominance. Two different concepts, but there is a narrow margin in explaining the two differences between them. On sex-linked disorders, please emphasize on how we are writing this. This is a female, and this female have a capital letter and a small letter, so she have both alleles on the chromosomes, but this one have just only one allele because it's a male and only on the X chromosome. They must only write it on the X chromosome. So now maybe you will sit and say, but we know that, but believe me, in the exams, they write that one on the Y also, and they are very confused on this one. If we look at this, please use the letters that the question gives you. So we are used to sex-linked disorder, in this case H for hemophilia, but sometimes they use another letter in the exam. So focus on what is the letter that the exam paper gives us to work with, and we use that one and not our own letters, because then the whole diagram is wrong. Nothing will be allocated marks. We must use as indicated in the exam paper, and the exam paper will indicate which letter you use. But how to write sex-linked disorder is very important. On this one, again, the alleles A, B, and E are different combinations that results in 4 blood groups. So there is 4 blood groups, A, B, and O. But the alleles, there's only an allele, A, a B, and a small letter I allele for blood groups. And this is how we write the different blood groups. So the learners are also struggling in writing the correct way, the blood groups in the correct way. Remember now, the O blood group is a recessive blood group, and this is the way we are writing it down and expressing it. AB is a codominance, where codominance is indicated. But again, just the way of writing blood groups are very, very important, and the way we write disorders are very important. Hemophilia, that is the male, it's a recessive allele, and this is a carrier, that is also a recessive allele. Just to show you. And then the typical questions that I ask on blood groups. You must be able to identify which one is adopted or maybe not a child. In this case, the father is A, the mother is AB, the daughter is A, and then they have two sons, one O and one B. So in this case, which one is adopted? Which one of the sons is adopted? It's son 1, because the father is A and the mother is B, and now he's O. That is impossible, because he must receive a recessive allele from the mother and one from the father, but the mother doesn't have one. You can see that the mother doesn't have one. So they must be able to identify these blood groups. So on blood groups, remember, blood groups can definitely say who is not the father. But blood groups cannot say who is definitely the father, because many males do have the same blood group. Blood groups cannot say who is definitely the father, but it can definitely say who is not the father. Just something there to remember. So now they ask us how many different phenotypes are there. There are 4. How many different genotypes are there? There are 1. How many different alleles do blood groups have? 3. And they can ask what is the given, or give the genotype of the father, and then you must be able to write the father's genotype. And there is the father's genotype. All right. Same in this case, which babies are swapped. If we look at this one, this is a baby, the mother and the father, and the baby is A. Seems right, because you can have the A from the father. In this case, the baby is O. It's not true. So this mother cannot have an O baby. And also in this case, this mother cannot have an AB baby. So these two babies are swapped. So just to show you how do they ask or evaluate blood groups in the genetics in paper 2. Different questions on that one. Also, diabreed crossing, just another way to ask it. The law, or also in genetics, that makes genetics very difficult, is the interpretation of a pedigree diagram. And then you must remember they don't have to indicate that the squares are males or the circles are females. You must know this, that squares indicates males and circles indicate females. Then another thing, you have to identify phenotypes of individuals when they give a genetic or pedigree diagram, or you must be able to identify a genotype or state which is inherited from each parent in this case of a genetic diagram. So just a tip on genetics. When the two are different in color, this one is not colored in and this one is colored in, we see that this one is female with brown eyes. We know brown eyes is a dominant allele. So in this case, there will be one that is homozygous recessive and one is heterozygous dominant. So if you get one with a color in and not color in, then definitely one will be heterozygous dominant and the other one will be homozygous recessive. So let's look at this one. In this case, clear and colored in. So one of them is homozygous recessive and one of them is heterozygous dominant. All right. So typical questions that they ask when they give a periodic diagram is how many generations are there, or they can ask how many males is there in this one. So how many males? 1, 2, 3, 4, 5, 6. 7, 8, 9, 10, 11, 12 males. Or they can ask how many children do this generation have. So there's a child, 1, 2, 3, 4, 5, 6. So how many children? 1, 2, 3, 4, 5, 6. Those are not a child, and that one is also not a child, and that one is not a child. Okay. So then they can ask what is the genotype and the phenotype, and you have to explain the genotype and the phenotype when you read through it. When you do this, make sure that the learners establish, is this a sex-linked disorder, or is it just an autosome link example, and they must identify. They must use the right way in writing it. So again, in this case, look at this. We don't give a key, so we can ask how many males. Then you have to know that males is squares, 1, 2, 3. Again, this one is not colored in. This one is colored in. So one of them must be homozygous, and the other one is heterozygous dominant. That is why we have, in this case, have the two offsprings. In this case, the offsprings are not colored in, but they do have a colored in offspring. In this case, both of them must be heterozygous, because this one is deaf, this person is deaf, and deaf is a recessive allele. So you receive one from the father and one from the mother. That makes them heterozygous and heterozygous. So interpretation of a pedigree diagram, always very important. And the pedigree diagram can count between 9 and 13 or 14 points. So if you don't know how to interpret it, it becomes very difficult to answer this question, and the learners get confused on what is the question, or what must they answer. And they must differentiate between the phenotype, is that what they can see, and the genotype, it's genetic makeup of gene component. So very important when we do periodic diagrams. They can do a lot of them to practice. There's no standardized one, and that one is always asked. They differ from each question paper to the next one. So a lot of these periodic diagrams are available in past exam papers. Please make sure that you practice it so that the learners can be able to answer the questions on pedigree diagrams. There's another one to show you. In this case, this one have a colored in child, so these two must be heterozygous. They must be heterozygous. Also, these two must be heterozygous because this one is colored in. So that is the tip that I have to you. Now look, explain. They must explain sometimes. They must explain why this one is colored in or have the recessive allele, and then they must explain it they get it from one allele from the father and one allele from the mother. Always very, very important to note. Then again on genetics, we must be able to state that genetics, also the fact of mutation and chromosomal mutation. So In genetics, we have mutation, which is a sudden change in the genetic composition of an organism. And that is the general explanation for a mutation. But we divide mutations into gene mutation and chromosomal mutation. Now remember, mutation is also a source of variation. So when we do evolution, we will come back to mutations or emphasize this again, because the learners must identify that what is a gene mutation? It's a change in the sequence of nitrogen bases of nucleotides in a DNA. And this is stated in the exam guidelines. So state it like that a gene mutation is a change in the sequence of nitrogen bases or nucleotides in DNA. And a chromosomal mutation is a change in the normal structure of the number of chromosomes. So under mutations, we have a gene mutation and we have a chromosomal mutation. Both of them very, very important in this case when we explain mutations. Then the effect of mutations is something completely different. The effect is that we have mutations that are harmful, we have mutations that are harmless, or we have mutations that is useful mutations. So that is the effect of mutations. So this is the different mutations, and that is the effect of a mutation. So differentiate between these two very carefully. So the last one on paper 1 is genetics, genetic engineering. So when we talk about genetic engineering, we're going to emphasize on the fact that genetic engineering is when we take one gene from one organism, we put it in another one's organism, and we change the genetic makeup of that organism. So we take a gene from one organism, we put it in another organism, and we change the genetic makeup of that organism. So you can decide, or is this a banana fish or a fish banana? And also you have to state, when we do genetic engineering, what is the disadvantages and what is the advantages of genetic engineering. In this case, maybe the advantage is with one bite you can have protein and carbohydrates or whatever, or you can also look at the example that I give. Usually there is a paragraph that will come with this type of question, and then disadvantages, so it is expensive. Research money could be used for other needs. It interferes with nature. It's immoral, or you can't play God. There is also a potential health impact, or we are unsure of the long-term effect on nature. So any one of those that you can apply on the example that is given. And also, this is also, you can make any exam paper if you question paper 2. That question paper 2 will have a question on genetic engineering. Always have a question on genetic engineering. Okay, and then for advantages, production of medication, just be sensitive for, we cannot say that, for example, if we use this one of bananas, that it produces medicine. In this case, it doesn't produce medicine. The advantages of genetic engineering: control pests, using specific genes to increase crop yield, selecting genes to increase shelf life of plant production. Now, please be sensitive. You must read the paragraph. You must read what is the paragraph giving us to work with. And in the paragraph, it will state the advantages or the disadvantages. So sometimes you must just read your paragraph very carefully and look at it so that you'll be able to answer the questions. So maybe in this case, the paragraph will say that the fish have a genetic gene that helps promote memories, to promote that you don't lose a memory loss, and then it's totally something, the advantages will be totally different. So then the argument will maybe be people don't eat fish easily, so they eat bananas. Or people in inlands don't have the advantage of fish, ocean nearby to catch fish or eat fish, but they have bananas and therefore they produce the banana fish for that specific advantage. So just read through the paragraph very carefully and stated the disadvantages and the advantages of genetic engineering. So the same one on genetic engineering, I will focus when we come to artificial selection at evolution. So what is the differences and the differences between genetic engineering and natural selection or artificial selection? So we will come to that, or what is the comparison between them. But in evolution, we will also look at that. But just remember that genetic engineering is the same as artificial selection, where humans decide which gene they choose to benefit human needs. So on that one is my last that I want to say on key points on paper 2. I know I said a lot, but there's really a lot of technical points and technical ways in how learners must explain their question or their answers, and you must please focus on that so that they can see to it that I do it on the right way, so to allocate some marks. Sometimes it's a very common answer, a common question and a common answer, but then the learners lose marks because their wording or the correct approach to the answer is totally wrong. So be sensitive on that. So from my side, on key points on paper 2, on genetics and DNA, that concludes this. I'm just going to take a break now, and then we will come back for evolution. And on evolution, it's then human evolution and evolution theories in general that we will look at and focus on the way they ask it and the way the learners must answer it. So thank you for this. Good afternoon. Let's start then on evolution. That is also a large part of question paper 2 that you must be able to answer. I'm going to use the slides. Most of you will know this slide. It's the previous Ronel Pretorius did make this slide. It's just a small adjustments on some of them, but you will see teaching evolution according to the guidelines. We have to differentiate between a hypothesis and a theory, and we have to differentiate between evolution and biological evolution. So when we look at the terminology, always there is a hypothesis and always there is a theory, and the theory is an observation. When we talk about the hypothesis, it's a testable statement. Now these two are always in a question paper. Either to give as a one word the biological term, or maybe to state the hypothesis or explain what is a theory. And two that we added here is extinction and biodiversity. And the reason for that is because extinction and biodiversity you did when you are in grade 10 with life science grade 10. So we can also evaluate this. So just look at extinction and biodiversity. So another two very important terminologies is continuous variation and discontinuous variation, where we have the variations show intermediate phenotypes like height. It's continuous variation, and discontinuous variation is where the variation with no intermediate phenotypes. These two I will show you on the next slide is the definition of a species and a population. Two very important terms: population, same species in the defined area at a given time, and then the species is where we have two species. If they can interbreed, they can produce fertile offsprings. Just want to emphasize here on interbreed. On many occasions, as you will see later on when we talk about speciation and natural selection, we're going to say we bring them together and interbreed and produce fertile offsprings. Be very sensitive not to say we mate them. They can mate, but they cannot interbreed. So to mate don't the learners use the word mate, but please stay away from that description. We're talking about interbreed and they produce fertile offspring. So why are they not a species? Because they can interbreed and produce infertile offsprings, then they are a species. So a very, very important concept in evolution: species and populations. Please emphasize on that one. So typical exam questions. Descent with modification over time is evolution. Example of discontinuous variation in humans is both of them, skin color and height, is they are continuous. Skin color and height is continuous variation. So in this case, none of them. A population with no intermediate phenotypes, that is discontinuous variation. So very important, discontinuous and continuous variation. Be able to differentiate between a hypothesis and a theory. That type of questions on the definitions. Total disappearance of a species from Earth is extinction. The variety of life forms is biodiversity. And a trait has a range of phenotypes is continuous variation. That is the different ways that they evaluate it. All right. Again, a lot of questions that I can answer and possible ways they ask these definitions. Again, in this stage, produce fertile offsprings, A and C. B and C produce infertile offsprings. So A and B are definitely the same species because they produce fertile offsprings. Just another way they can ask it. Evidence of evolution, very important. We have fossil records, we have biogeographic, we have homologous structure, and we have genetics. Fossils, explanation of fossils, explanation of genetics. Homologous chromosomes, remember the build are the same, but the function difference. That is homologous chromosomes. And then cultural evidence is the use of tools and weapons. And we also have biogeographic, which stated that there is they developed from a common ancestral. These two different species or examples that they can use to explain that. Typical questions, present day distribution is biogeographic organisms. Evidence of evolution, that is the way they ask it in the exam. Then this one about a variation, very, very important. What is the sources of variation? Describe how each of the following contributes to variation among individuals. Meiosis, I explained meiosis in crossing over and random arrangements in the first part. Also mutations, I did explain mutations. We have a chromosome mutation and a gene mutation. And then the other sources are chance fertilization. Any female or any sperm cell can fertilize any egg cell, and random mating is where any male and any female can mate. So that is very important, two very important sources of variation. So we have meiosis, and then chance fertilization or random arrangement, or chance fertilization and then mutation. Remember, random arrangement and crossing over form part of meiosis. So if you have to explain the sources or name the sources, rather name meiosis and then chance and random fertilization and then mutations. Meiosis, mutation, chance fertilization, random mating. So that is the 1, 2, 3, 4. So if you have to name it, meiosis separately. It's not meiosis, crossing over and random arrangement. Crossing over and random arrangement form part of meiosis. So you will have a mark for meiosis, but these will be wrong, and then they will lose marks. So if you only have to name the sources, it's meiosis, mutation, chance fertilization, and random mating. Crossing over, explain that to you in genetics. Random arrangement, also explain that to you. What is random fertilization? Different egg cells and different sperm cells, and mating between organisms with a species lead to a different set of offspring. So where any female and any male can mate with one another. Alright, and mutations, we have gene mutation and chromosome mutation, which are very, very important. Again, what is the difference between gene mutation and chromosome mutation? As I mentioned it with genetics earlier, the variation and the definition of a gene mutation and the definition of a chromosome mutation is very, very, very important, and they must be able to explain it in such a way. Just another typical question that they can ask. Cloning is not a source of variation because it's an identical copy. The organism makes an identical copy there. Meiosis is also not a source of variation because the chromosome, the DNA is the same. State what is meant by artificial selection and natural selection. Tabulate differences between natural selection and artificial selection. Very, very important that we know the differences on this one. And I want to go to this particular slide where we tabulate the differences between natural selection and artificial selection. So they can either ask what is artificial selection, and then you state it's a human, that is the selective force, responds to satisfy human need and can include more than one species. Or what is natural selection, then you state that. Or a very favorable question is tabulate the differences between natural selection and artificial selection. So then we indicate environment is the selective force. Humans are the selective force. In the environment, the one with the favorable characteristic will survive. So it's a response to the adaptations to nature. And this one is a response to satisfy human need. Artificial selection include more than one species, and it happens in a species. In this case, I just want to emphasize the fact there was always a difference, like a natural selection is a long process and artificial selection is a short process. We try to stay away from that differences or explanation. So it doesn't go good with DBE. Long process and short process. So leave that one out in the differences between natural selection and artificial selection. But as I stated, a very favorable question, always asked in an exam paper, or you must give the definition of artificial selection, of natural selection. But natural selection will come to natural selection when we do the theory of Darwin also. So the characteristics of an organism can be changed through selective breeding and the genetic engineering process. Name two similarities between selective breeding and genetic engineering. Remember, that is humans is the selective force, and it's to satisfy human needs. So, and then there's explain two reasons why some people might be against the use of genetic engineering, and that one is for it's expensive, it is immoral, we don't know what is the health impact, we don't know what is the long-term and effect on the environment, etc. So there is the answers by humans and to satisfy human needs. It is not necessarily benefiting for the organism. It's another one on what is the difference or what is the similarities between artificial selection and genetic engineering. Again, a lot of questions on state two benefits of selecting breeding of chickens other than for increasing meat production. Typical questions on genetic engineering, but remember now selective breeding is also in evolution, and that is why these questions are complementary one another in paper 2. A lot of questions on artificial selection, so that the learners must know what it is and what is the difference between them. So now on the evolution theories of Lamarckism, Darwinism, and punctuated equilibrium. Now this is very important, and it's also evaluated in paper 2 a lot of times. The answer in many times of these questions is the same. They just use other examples that you must use to apply your knowledge of Darwinism and your knowledge of Lamarckism to that example, but we will come to that in a moment. But also, please make sure that the learners can spell Lamarckism and Darwinism and punctuated equilibrium, because it's many times asked as give the biological term for that. So if we look at Lamarckism, the theories of evolution of Lamarck, and you have two theories, the law of use and disuse, and the law of inheritance, and you must explain these two laws. So the laws, it's actually, when you use it, it becomes bigger and better, and if you don't use it, it becomes disintegrated. And this inheritance of acquired characteristic we can carry over to the next generation, which is totally not true in case of Lamarck's theory. So the learners must be able to state the two laws and give the explanation of the two laws. Very important in evolution. Then Lamarck's explanation. Sometimes you have to explain according to Lamarck, and this is the old way, and it's totally correct. What was the original characteristic at the start? What did the organism do? Why did the organism do this? What was the result at the end? And what happens to this new characteristics? And then the learner have to take the example and apply the example to the guiding questions. So this must be practiced at school and in class, where you have a lot of examples and explain. They must explain Lamarck's theory on a certain way, in a certain way, on a certain example. So maybe in the future we will have a question on why humans have grow mask on their face. Explain how, according to Lamarck, how did this happen? And then your answer will be, what was the original characteristic in the start? No, humans did not wear mask. What did the organisms do? They wear masks because of the coronavirus for more than 2 years. So they prevent inhaling the coronavirus. What was the result? They start to grow a mask on their face. What happens to this characteristic? Babies that was born after 2023 were born with a mask on it, and that is according to Lamarck. So what was the original characteristic? What did the organism do? Why did the organism do it, and what is the result? So we acquire a mask, so we wear a mask every day, and that makes us to grow a mask, and that is the acquired characteristics that we passed on to the next generation. So according to Lamarck's theory, just an example. So they can use any type of examples, and that is the guiding questions that we must apply to that question. So you know, all of you know this typical example of the cacti plants with long roots and short roots. So no one have long roots, but they want long roots, so they grow it to absorb water, and the offsprings, then they become long, and then the offsprings have this acquired characteristic. So as simple as that. But the main thing is we must apply the guiding questions on as much as possible examples that you have. Now also in my gut feeling I have this one of Lamarck, because all the November exam papers, they will ask natural selection according to Darwin, not so much on Lamarck. So somewhere, if it is not this year, maybe next year they will evaluate the Lamarck's theory on acquired characteristics. So the reason why we are not accepting this, acquired characteristics are not inherited. Organisms did not evolve because they want to evolve. So we can have masks just because we wear them all the time, or we want them, and then we grow a mask. So Lamarck's belief in determination is wrong. All right. So that is why we're not accepting Lamarck. Then we are with Darwin's theories. Remember the one that is stipulated in the exam guidelines on Darwin's theories of natural selection. It is the general one. This is the general explanation of natural selection. So if you are lucky, they can ask it like that, straightforward. Describe Darwin's theory of evolution by natural selection. And then we have to identify that there is also a variation. We have favorable characteristics and we have unfavorable characteristics, but that is the general answer on natural selection according to Darwin. But usually in Darwin's case, they will give us an example, and then we have to use the guiding questions on that one to explain the example that they give. Now, believe me, since the one of the bongi, remember, did you remember the bongi bok, that one? I think it was 2016. Since then, each time they try to out-compete the previous year example with the most abstract type of example of natural selection. And again, it will happen like that on natural selection. Also on speciation, they have this weird, unfamiliar example, so it will be there again. But there's actually two things that we have to remember when we talk about natural selection according to Darwin. The first one is just simply identify the variation. So let me just go back to an example. So just identify the variation. In this case, long roots, short roots. So that is, state the difference and state what the difference is. There is the plants have long roots and they have short roots. So now you establish a variation. The other thing that you must trying to identify in the example is what is favorable and what is unfavorable. And that is all actually with natural selection. If you can drill this into your learners, establish the variation, then they must explain what is the favorable characteristic. And remember, the favorable characteristic always survive and why. And the unfavorable characteristic always die and why do they die. And then this favorable characteristic is then carried over to the next generation. But it's very important to name the favorable characteristic. In this case, the favorable characteristic is long roots. Why? They can absorb water and therefore they survive. Short roots cannot absorb the water and they die. And now the favorable characteristic of long roots is then carried over to the next generation. So actually natural selection, any example that you find, identify the variation, what's favorable and what's unfavorable, and then you can answer your question, irrespectively what the example is. For example, then it was the bongo bok or bongo that was living in the thick bushes of Africa. So it's a very unfamiliar example, but the variation was some of them could lay back their horns and some of them could not lay back their horns. So the one that could lay back the horns was the favorable characteristic. They survived because they didn't catch up in the thick bushes. And the one that could not lay down their horns, they was caught up in the shrubs and the bushes, and the predators ate them and they died. So the favorable characteristic was horns that can lay back, and that was carried over to the next generation. So be very sensitive on the example that they give, but really in the example, in the explanation, in the paragraph, it will clearly indicate what is favorable, what is unfavorable, and it will clearly indicate the variation that they're talking about. So when explaining natural selection, usually it counts 6 marks. You state the variation, you state the favorable characteristic, you state the unfavorable characteristic, you state that the favorable survive and why, and the unfavorable die and why, and then the next generation will have the favorable characteristic. So in another way, natural selection can also be evaluated by giving an example like this. Maybe then they state, what is this? They ask, what is this phenomena? So you must state that this is natural selection. What is happening here? Natural selection. Which is the favorable color? In this case, it's white. which is the selective force? Nature is the selective force. Why is it white color that survived? The predator could not see them, so they survived. The black ones was exposed, and they were not camouflaged, so the predators catch them. So again, state the favorable, state the unfavorable character. This one survived, this one died, and the reason why it is. So natural selection, always in a question paper for 6 marks, state natural selection according to Darwin. You refer to the variation and state the variation. In this case, there's a variation. There's white mouse and there's black mouse. And what is the differences? Oh, that is the variation. Which one is the favorable characteristic and which one is the unfavorable characteristic? So please prepare your learners that the example will be a bit extreme or a bit not used to or familiar, but they must just read and identify the variation, also identify the favorable and the unfavorable characteristic, and then they will be able to answer the question. Differences between Lamarck and Darwin's theory. There's the differences also. Once there was a question like this in March in a supplementary exam in March I think it was 2018 or 2017, that I found this question asked. Maybe we must prepare ourselves that they can evaluate this one again, because it was a long time that I found this question just in one question paper when I do some research on questions for paper 2. So maybe your level 1 or level 7 learners can prepare them for a differentiation or differences between Lamarck and Darwin as stipulated in this table. Then example questions again of variations and survival in a habitat, as I said before. Identify the variation and identify the favorable characteristic and the unfavorable characteristic. So there is a question. So they ask, explain natural selection. So what is the variation? Which is the favorable characteristic? Which is the unfavorable characteristic? Which one survive and which one die again? All right. Another one of examples, and also one of male guppies have bright colored spots to attack females, but these spots also attract predators. It was previously observed that males living in a stream where there were many predatory fish tend to have fewer spots to reduce their risk of being eaten. So according to Darwin's law of natural selection, there was male guppies with lots of spots and male guppies with less spots. So that is the variation. So the less spots survive because they won't be eaten by predators or they won't be spotted by predators. Those were and they survive. And the male guppies with a lot of spots were easily seen by predators. They were eaten by predators, so they did not survive. So the favorable characteristic of have less spots are carried over to the next generation. Variation, favorable and unfavorable. State what happens to the favorable and what happens to the unfavorable and why. And then you will be able to answer the question in the exam paper by just applying. You seek the variation and you state the variation very clearly in full sentence. What happens to the favorable characteristic? What is the favorable characteristic? What happens to the favorable characteristics? And it survives. And the unfavorable characteristic, what happens to it? And it dies. And the favorable characteristic. And mention then again, what is the favorable characteristic? In this case, less spots will carry over to the next generation. So there is a lot of that. And then we come to equilibrium, punctuated equilibrium. Very important in punctuated equilibrium to indicate or to state that it is evolution where there is long periods of time where species do not change. And then there is short periods of time where rapid change occur through natural selection. That is punctuated equilibrium. And the two persons that are responsible for the formulation of this model was Gold and Elrich. And they must be able to identify Gold and Elrich. The names, not the persons, but they have to identify that Gold and Elrich was the ones that formulated the equilibrium model. On this also, remember now there's a few names in life science, question paper 2, that we must remember. We must know that Gregor Mendel is the father of genetics. Charles Darwin is the father of evolution. Watson and Crick was the two persons that first named the double helix structure, and then of course Gold and Elrich that formulates the punctuated equilibrium. Okay? So what does equilibrium, this punctuated equilibrium stands? This is periods of change, alternative with short periods where rapid change might take place. That is punctuated equilibrium. Either they can give you a graph just to show you that this is gradual evolution taking place, and this is punctuated, long periods there is no change, but then there is a short period with a rapid change on the color or the species or whatever. Just a diagram or graph to explain it. No change, rapid change. Long periods, there is no change, then there is a rapid change again. Right, I want to take a time on the formation of a new species, which is called speciation. Now, first of all, the learners get confused on the format and the way of answering speciation and the way of answering natural selection. Especially if they ask both of them in a question. Question paper 2, markers at question paper 2 will also tell you it's very difficult when sometimes speciation and natural selection is in the same questions, usually question 3, then also markers get confused with the sequence in marking natural selection and speciation. So try to emphasize that there's a difference in the way we answer natural selection, and there's a difference in the way we answer speciation. And there's a sequence when we answer it. When we answer natural selection, we state the variation, we state then the favorable characteristics, what happens to it, why, then the unfavorable, what happens and why, and then the fact that we are carrying over the favorable characteristics. So a few technical points in speciation that is very, very, very important when answering speciation. Let me just go to the yellow slide, and this is straight from the new exam guidelines. And we start with, if a population of a single species, I just want to stop there. Now you take this and you apply it to the question that they ask, the example of the question that is asked. If a population of a single species, whatever the question is, maybe the question is on long-tailed birds, or the question is on tortoises, or whatever the question may be, you use the question's example and you state if a population of a single species, whatever the species name is, becomes separated by a geographical barrier, and then you must mention the type of geographical barrier. Now, in the previous exam, this two-sentence, this full two-sentence, they allocated 1 mark for that. But you have to state if a population, the original population, or the single species. Remember now, the speciation started from a single species, a population of a single species, not just population, but this population of a single species and what the example is. Now, then the population is split into 2. It's not necessarily 2. It can be 2 or 3 or 4. So the learner must read or look at the diagram and identify in how many ways this population splits. If it splits, after it splits, there is no gene flow between the two populations. There's no gene flow, so the original population splits into 2, and there are no gene flow between these two populations now. And the sequence must be there, like this. Now, since each population may be exposed to different environment conditions, natural selection occurs independently on each of the two populations. So now, back to this one, the population splits into 2. Either this will be now on the left-hand side of a mountain and the right-hand side of the mountain, or of a northern part or from the southern part. So state it there. State where it is divided into. They are split into 2 populations on an island or on 2 different mountains, or whatever the example may be. So I'm now, after this I will come to an example so that you can see how we explain it. Since each population may be expected different environmental conditions, maybe in the example they will state the different environmental conditions. They will say the northern islands are very cold and the southern islands are very warm, and then you state this and you say they are exposed to different environmental conditions. The population on the northern side are very cold, the population on the southern side is very warm. So you apply these general points to the example that you are going to answer. Then natural selection occurs independently in each of the 2 populations. Maybe they will say the northern natural selection, those in the northern part died from the cold. Then you explain the selection force. Maybe there's a predator or whatever. Such that the individuals of the 2 populations become very different, and they become different genotypically and phenotypically. So somewhere they will maybe explain the 2 differences, and they become genotypically and phenotypically, ones with thick hair in the northern parts where it's cold, and one with a thin layer of hairs in the southern part. So the description, the phenotypical description, you can also mention the difference in the phenotypical of the 2 species that is separated now. Even if the 2 populations, now you must say, even if the 2 populations, what is the 2 populations now? The 2 populations of long-tailed feathers in the northern part and short-tailed feathers or long hair bears in the northern part and short hair bears in the southern part were mixed again. So you mention now the phenotypical difference of this population, and you mention the two populations that is coming back together again. They will not be able to interbreed. Please continue with and produce fertile offsprings, and the two populations are now different species, one in the north and one in the south. So I think I said a lot of things now here, but let's look at the example, and then maybe we can get clarity on this one. So the differences, we must explain that original species. We must explain the geographical barrier. Which one is the geographical barrier? In how many populations does it split into? There are different environmental conditions, natural selection independently. There are different species now. Maybe there are two different, for example, the emos and the ostrich. There are two different species now. There are emos and ostriches, and they different genotypically and phenotypically. The ostrich is larger and have black and white feathers, and the emo is smaller. Even if the two populations were mixed together, even if the emo and the ostrich are mixed together, they cannot interbreed and produce fertile offsprings. The two populations are two different species now, namely ostrich and lemurs. So, if we look at that example, the original population of flightless birds becomes separated by geographical barrier, the sea. Then the population was split into three. Two, the emo and the ostrich, and there was no gene flow between the two populations. Each one experienced different environmental conditions. Natural selection is independently, whatever the natural selection may be there on each island, and they are phenotypically and genotypically different. Two populations were mixed together again. When we bring the emo and the ostrich together again, they cannot interbreed and produce fertile offsprings, and they become separate species now, namely emos and ostrich. Very important. Speciation, very, very important, and you must apply it to an exact example. So, let's look at this one and try to explain it in the new way that they want us to explain it. So we started with this original population of tortoises. Species A was the original population of a single species of tortoises. They were split by a geographical barrier, which is the sea, into three different islands. Each of these populations on these different island 1, 2 and 3 undergoes different environmental conditions. They undergo natural selection independently on island 1, 2 and 3. Now the tortoises are different, and they differ phenotypically from one another. A is the original one, but B differs from them, phenotypical and genotypical, and C differs also from A, phenotypical and genotypical. And when we bring them back together again, they cannot interbreed, they cannot produce fertile offsprings, and in this case we have different species A, B and C. And the mark allocation in the new way of answering it, it's 7 to 8 marks for speciation. apply the speciation as stated in exam guidelines. Apply that to your question. And at the end we must say it's now different species and you name the two different species. Usually they will give you an example and they will say there was species, original species, and how did the original species looks like, and now there are two different because the other one on the other side of the mountain maybe have red feathers and this one have white feathers, and then you have to explain how speciation takes place. Alright, these are just another one. So there's a geographical barrier of a rocky island. So that is the geographical barrier. They show variation in species in which fish living in a lake. There was a rocky island that extended across the whole length of the lake, but it was underwater due to high water levels. The fish were therefore able to move freely throughout the lake. Diagram 2 shows the same lake many years later. A drought had caused the level of the water of the lake to drop, so that the rocky island in the center separated the lake into smaller lakes, A and B, for a long period of time. So you can see now what is the fish population, and they asked the definition of population. State 4 possible causes of variation: meiosis, mutations, random fertilization, random mating. Name the process that result in the fish population become two different. The speciation, and now they say explain how the new species was formed. So if we look at this, we're going to this diagram and say in diagram 1 the original population of fish species were separated by a geographical barrier, a rocky island, into two different populations. There were no gene flow between them. Each one undergo environmental conditions separately, or each one have experienced different environmental conditions, and they undergoes natural selection independently on each lake A and lake B. So now the fish become genotypically and phenotypically different from one another. Those of them don't have spots, those have spots and stripes, and the two populations like A and B are different from one another. If we bring them together again, they cannot interbreed and produce fertile offsprings, so they are different species. Like A and like B have different species. Alright, there's another one on speciation, and then just to show you reproductive isolation mechanisms, be sensitive to the fact that I can give you an example and then you have to identify the type of barrier. So maybe they say the season of breeding of species B and species A are different. What type of isolation mechanism is that? Reproductive isolation mechanism, and then it's breeding at different times of the year. Or they can show you a picture, or they can tell you in a paragraph that a bird is singing to another bird, and that is a mating call. And what is the isolation mechanism? Then it's species-specific courtship behavior. So either you must name the barriers, or you have to identify the specific isolation, reproductive isolation mechanism. So if you learn this, apply some examples and of what is species-specific courtship and what is breeding at different times, so that learners can identify it and name the isolation mechanism. Alright. So that is teaching evolution in general. So let's look at evolution, human evolution. The one on human evolution. The evolution is 65 marks, irrespectively human evolution or general evolution. But in human evolution, the question stays the same, but the diagrams maybe will different. And the questions that is usually asked in human evolution is, we must be able to identify that Hominidae is the family name for all primates. Hominidae is the family name for all primate species, and the spelling of Hominidae. Hominidae is the family to which all primates belong. Then the phylogenetic tree, always a phylogenetic tree in evolution, always evaluated in evolution, and the phylogenetic tree is where we look at the evolutionary relationship among a set of organisms or a group of organisms. So a phylogenetic tree, you must give the definition. We must identify what is the phylogenetic tree. In this case, it shows the timeline, and there's the Hominidae. All the primates are belonging to the Hominidae. Just the difference between Hominidae and Homini, Homin and Homid. If you see a species referred to as a hominin, it means it belongs directly to the human line, whereas something refers to as a hominid could refer any one of the great apes or humans. So hominid is humans and apes. Only hominin is humans, only humans. Alright, so when we look on a phylogenetic tree, it's very important to understand the phylogenetic tree. So this is the ancestor for 1, 2, 3, and 4. That is the ancestor. Speciation happens here. B and C are more closely related, and they share a more common ancestor. So again, this is the common ancestor of A, B, and C. This one is the common ancestor of B and C, and they are more related to one another. If we look at the diagram like this, I want to emphasize a few things. There is a lot of questions that we can ask on a phylogenetic. There's a lot of questions that we can ask on a phylogenetic tree. So first of all, this diagram represents a phylogenetic tree. A. ramidicus is the ancestor of all these species. So a phylogenetic tree. In this case, how many species are in this phylogenetic tree? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. There are 14 species. Next question will be, how many genuses are in this phylogenetic tree? Homo, H is for homo. There's a homo, 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 homo. And then there's the P, so that is number 2. And then there's the A for Australopithecus, number 3. And Ardipithecus is number 4. So in this case, we have 4 genuses. This is a phylogenetic tree. How many species? How many genuses? Other one that I always ask is, which fossil is considered to be the first one to use tools? Homo habilis. Which two are very closely related to one another? P. robustus and P. bossii, because they have a more recent common ancestor. Those two are also related to one another, most related to one another because they have a common ancestor. So in this case, fossils, how many, or name the Australopithecus that is only found in Africa, then we must name Karabo, Mrs. Place, Taung Child, and Littlefoot. How many years ago did Ardipithecus ramidicus evolved? 4.5 million years ago. So a lot of questions that can be asked on this one. What is the differences between Homo sapiensis and, for example, Australopithecus afarensis, and the similarities, but we will come to that later on. But just giving a phylogenetic tree, on this one we can ask a lot of questions on a phylogenetic tree. What does a phylogenetic tree represent? Possible evolutionary relationships. The family to which homins belongs to, Hominidae. Always asking that. And to interpret the phylogenetic tree, again, a phylogenetic tree, this diagram is a phylogenetic tree. It has 1, 2, 3, 4, 5, 6, 7 species. And how many genuses they can ask you, and you have to answer that. Which one is the fossil that is used tools? Is always Homo habilis. Which one is the direct ancestor of Homo sapiens? Is Homo erectus. So a lot of questions on phylogenetic trees. How many of each of the following are represented in this diagram? Genera, species. Explain why I. robustus and I. bossii are more closely related. If we look at that one, why are they most closely related? Because they share a most recent common ancestor. They have a most recent common ancestor. And that's the type of questions that we ask. Explain how the location and the age of Homo fossils are used as evidence of the out of Africa hypothesis. When we come to Africa hypothesis, I will emphasize something there to you also. Which one first to use tools? Homo habilis. Name 2 Australopithecus found in South Africa. Karabo, Lucy, sorry, not Lucy, Mrs. Place, Littlefoot, but I will give that later on to you. So a typical questions, the diagram might differ, but the question stays the same. Again, phylogenetic tree, identify 2 bipedal organisms. How long after developing the ability to walk on 2 feet did pre-humans develop? And you take your phylogenetic tree and you can read all the answers. So there is bipedalism. Those are those that use tools. And you can interpret the phylogenetic tree. Right. Then, list characteristics that humans share with African apes. Also, tabulated differences. Typical questions. Favorable questions, questions that we always ask. So there's the list of characteristics. So please make sure the learners knows the whole list, not just 3 or 4, because they can ask only on the characteristics on the eyes or the long upper arms. Favorable one is name the characteristics that we share only on our upper arms. So we have long upper arms, freely rotating arms. We have elbow joints allowing rotation, flat nails, and then it's only on the upper arms. So all these all these we must be able to identify the characteristics we share with other primates. Yeah, that is what there's nothing there that I want to show. Okay. Precision grip is one of the differences because of the thumb. That one. Right. The ability of an organism to walk on two limbs is called bipedalism. The opening in the skull through which the spine cord enters is also the foramen magnum, but I will come to that one when I'm going to explain the differences between African apes and human. So in this case we must emphasize on the differences of the foramen magnum, the spine and the pelvic girdle. But these differences are on bipedalism. So very important in this case, what happens or what is the changes for bipedalism and what is the significance of this bipedalism. And then we're going to look at brain size, we're going to look at teeth, we're going to look at prognathism, palate shape, cranial ridges, brow ridges, and also on some very important technical points that is important when we do the differences on this. So let's start on the foramen magnum. So the foramen magnum, there is the foramen magnum, and in a sketch like this, more at the back, more in front under the skull. So the foramen magnum moves from more at the back to more in front underneath the skull. That is what we must write when we write on the difference on the foramen magnum. Again, more at the back of the skull to more in front of the skull, under the skull. That is the foramen magnum, and that is called the foramen magnum. So that is on the foramen magnum. The pelvis moves from a long and narrow to a wide and short pelvis. Long and narrow to a more short and wide pelvis. So I will come to the significance later on of these two, of what is that. The spine, we have a C shape and the S shape. That is the difference in the spine. The difference in the cranium or the skull is small to big. The cranium is small to big. I will say something on the brain just later on when we must use the brain, but in this case the skull or the cranium. So typical questions again. Evidence of human evolution. So the atomical differences between humans and African apes. So you can see I put there, I close brain on the table. When you have to differentiate between humans and African apes, many times I give two examples and then they ask, give the differences. If they give you a skeleton or a skull like this one and this one, then we don't use the brain as a difference because we can't see the brain when they gave that one. So stick to a large cranium and a small cranium. Brow ridges are not well developed. Humans does not have, many learners wrote no brow ridges. We do have brow ridges, it's not well developed. Brow ridges are well developed, African apes are well developed and not well developed. No brow ridges. Then the spine is straightforward, the pelvis straightforward. Then the canines, we have small canines and large canines, not short and long. It's small canines and large canines, and it's canines, and we spell canines like that, not a K and a 9. It's the word canines. So be attentive on that one, please. The palate shape is round and rectangular. I think that one is straight. Now the jaws. Now if you use small jaws and large jaws, you are not using then less pronounced and more pronounced. Small jaws, large jaws, less pronounced, more pronounced. I'm going to show you a picture of that to explain the protruding and more protruding and less protruding, and prognance and more prognance. Cranium ridges, no cranium ridges at the top of the cranium, foramen magnum, and in a forward position and in a backwards position below the skull. That one must be there, below the skull, so that you can indicate that you know that the foramen magnum is below the skull and in a more forward position and in a more backwards position. So remember now, not short canines and large, long canines, small and large. Now in this case, a flat face and a sloping face is the same as less protruding jaws and or more protruding jaws. So if you look sloping face, you can't say a difference is sloping face and more protruding jaws. That is the same concept. A sloping face have a more protruding jaw, and a flat face has a less protruding jaw. So that one. Same with a forward slope and a forward slope more backwards. So we'll use flat face and sloping face, then we know our wording and our definitions are correct. Then on this one, we have canines. There is gaps, that is also called diastemas. There's gaps between them, and there's no gaps between them. Not small, no. So the difference is there's spaces, no spaces. All right? And then the shape of it is rectangular and C-shape. And there's the intermediate one. The palate shape is curved, and the reason for that is to roll food in a bolus to swallow, and at apes it's flat and hard to tear and grind the food. So again, spaces, no spaces. Large canines, small canines. There's a typical question. They ask, what is this is called? That is called the foramen magnum. Make sure of the spelling of this. And then they will ask differences and which one is bipedal and which one is quadrupedal. So this one is, the foramen magnum is more at the back and it moves more forward in under the or below the skull. Small skull. So this one is the skull now. Small skull, large skull. That part is the cranium, but this one is a small skull and a large skull. That is the differences that they ask. Okay, the differences on the spine and the pelvic girdle. Again, differences. Explain the foramen magnum moves in a more forward position, and that is more to the back below the skull. Long and narrow, small, short and wide. That is the differences that we can see, and always be able to explain the differences or tabulate 3 visible differences, and explaining how the shape of the pelvis contributes to bipedalism. long and short, no, long and narrow to short and wide. Again, a diagram like this, you have to explain the differences, look at the foramen magnum, be able to label it, the differences in the jaws and the teeth, spaces, no spaces, large, small, rectangular and C shape. Same questions again. Now also we must emphasize on the fact what is the significance of the foramen magnum that was in a backwards position in ape-like beings and now is in a forward position in the modern humans. So the significance is increase awareness of the environment, free of the hand to use implements, exposure of a large surface area for thermoregulation, and we can display of sex organs. So that is everything on the significance on the foramen magnum. The position of the foramen magnum that moves more forward, that is what is the benefits of that one. And then the cranium, we are a cranium that are, the cranium is from small to large cranium, and the benefit is better coordination, processing of large amount of information, processing of information faster, development of spoken and written language to communicate. So the difference between the cranium and the skull, as I indicated here, this is the cranium, that part is the cranium, and this is the skull. So in this case you will refer to a small skull and a large skull. The small skull have a smaller brain and the large skull can have a larger brain. So cranium is just that part. Therefore, if you tabulate, use the word skull in the table and not cranium, because in the picture is a skull, the picture is not a cranium. So jaws, humans have small jaws. Humans have jaws that are non-prognance compared to the jaws of ape-likes, and C-shape and the U-shaped jaw. And what is the benefit of that, or the significance? It's the diet from hard and raw food in ape-like beings to softer and cooked food. So they tend to ask the significance on, especially on the foramen magnum and the cranium and the jaws. They love to ask that, what is the significance of that? Then the teeth, also no gaps. Humans have smaller teeth. And what is the significance? Therefore, they correspond with the decrease of need to bite and tear, and increased need to grind and chew in humans in view of the changes in diet to soft and cooked food. So that is the significance. So that was the differences, and what is the significance in that one. The same with the eyebrow ridges. What is the significance therefore? And also on the chin, it helps with speech. So state the difference, must be able to state the difference and explain the significance of that one. I think it's very important to make sure that the level 7 learners are able to identify the differences and then the significance on that. Not maybe they will ask it maybe on the pelvis, maybe on the foramen magnum. They won't ask all of them because that will allocate too many marks, but definitely maybe on two of the changes, maybe on foramen magnum and the pelvis or the cranium, and then you must explain the significance that is accompanied with that changes that is made. So the geometric arch, less developed and more developed. The last two times that they asked the differences between skulls, they acknowledged this one. They did acknowledge the differences on the geometric arch, but they are not favoring this difference. So I think emphasizing on the others are more important than this one. Right, Africa, the home of humanity. So we start with the hypothesis, the out of Africa hypothesis. Very, 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 very important. Now in this case, in previous question papers, they asked, explain the out of Africa hypothesis according or use fossil evidence to explain the out of Africa hypothesis, or use genetic evidence to explain the out of Africa hypothesis. So in this, I want to emphasize that they can now ask it separately. They can ask, explain Australopithecus. Fossils, name the fossils of Australopithecus that was only found in Africa, and why is that evidence of the out of Africa hypothesis. So Ardipithecus, found in Africa only. Ardipithecus, found in Africa only. Australopithecus, found in Africa only. But which Australopithecus is only found in Africa? And it's only Karabo, Littlefoot, Taung, Child, and Mrs. Priest. There isn't the other one that we learn that are found in Africa only. But only Ardipithecus. Only Australopithecus in Africa, including Karabo, Littlefoot, Taung, Child, and Mrs. Priest. So when you explain Australopithecus, why does the fossils of Australopithecus contribute to the out of Africa hypothesis? Is because they are only found in Africa, and the names are Karabo, Littlefoot, Taung, Child, and Mrs. Priest. Now it's Taung Child, not Taung Baby. It's Littlefoot, not small feet. It's Mrs. Priest, not Mr. Priest. So just a few things that you must be attend to when you're discussing the Australopithecus. Homo fossils, Homo habilis, found in Africa only. And then oldest fossils of Homo erectus and Homo sapiens is found in Africa. Not in Africa only, because there's other parts of the world that they found them. I think the Great Rift Valley, but only found in Africa was Homo habilis. Then the also oldest fossils of Homo erectus and Homo sapiens was found in Africa, the oldest fossils, which means that if they're the oldest, it was many years ago, million years ago, that they were found in Africa. And then they compare this, that the oldest one was found in Africa, and younger fossils were found in other parts of the world. That means the oldest fossils of Homo sapiens is found in Africa, and younger fossils, more recently fossils, were found in other parts of the world, meaning the fact that the oldest ones was in Africa means that they contribute to the out of Africa hypothesis. So if the oldest fossils are found in Africa, it means they first come from there. But there are others that was found in other parts of the world, but they were younger. So it sounds like a lot of information, but in this we are referring to homos. And when we refer to homos, we said Homo habilis was found in Africa only. Homo habilis wasn't found in other parts of the world. Homo erectus and Homo sapiensis were found in Africa, but the oldest one, while the younger fossils were found in other parts of the world. So that is the contribution of Homos on out of Africa hypothesis. This one is the contribution of Australopithecus on out of Africa, and this one is Ardipithecus that is found in Africa. So, evidence of the out of Africa hypothesis is fossil evidence, and the genetic evidence is mitochondrial DNA, and we will come to mitochondrial DNA later on. So there's the phylogenetic tree. I started with that. Interpretation of a phylogenetic tree. Right, again fossil evidence as stated in your exam guidelines. Just to make sure we know the differences of evidence. So we have evolution, we have fossil evidence, genetic evidence, comparative anatomy, that one of homologous organs, and biogeography, that is evidence for evolution. Human evolution, fossil evidence, genetic evidence, mitochondrial cultural evidence, tools and weapons, and also biogeography. And the out of Africa hypothesis, we have fossil evidence, we have genetic evidence, we have cultural evidence, and biogeographic evidence. But they can ask, explain fossil evidence as an out of Africa hypothesis, and genetic evidence as out of Africa, also these two. Then we have to explain. And when we explain fossil evidence, then we refer to Ardipithecus, Australopithecus, and the Homo species. And then the out of Africa hypothesis, when we have to explain mitochondrial DNA. Now, mitochondrial DNA is what happens here, that the ovum is fertilized by the sperm cell, but the sperm cell, only the sperm cell's head is fertilizing the ovum. He loses the neck and the tail. That means the mitochondriums of the male is not transferred to the female. So the new offspring doesn't have mitochondrial DNA from the father. It's lost. That is why we can use mitochondrial DNA to trace back Mother Eve. Because we can go from individual to individual and look into the mitochondrial DNAs, and then the mitochondrial DNAs will give us markers that can go, and we can trace it back to Mother Eve. So when we're explaining mitochondrial DNA, when you have to explain mitochondrial DNA or genetics as an out of Africa hypothesis, we just remember the following. Mitochondrial DNA of the mother have mutations that act as markers to trace back to Mother Eve. So there's the 5 M's. Mitochondrial DNA of the mother, mitochondrial 1, of the mother, 2, have mutations, 3, that act as markers, 4, that can be traced back to Mother Eve, number 5. Mitochondrial DNA of the mother have mutations that act as markers that we can trace back to Mother Eve. And that is how we explain mitochondrial DNA as evidence for the out of Africa hypothesis. Also, a very important part of evolution, very famous question to ask, and then the learners really struggle to get to the answer, as with the out of Africa hypothesis. So just another way, they ask, they can ask this question, and this is how they answer it in the exams. So, using DNA evidence, so this is the mitochondrial DNA that you must use to explain out of Africa hypothesis. A lot of questions you can do on that one. Apart from DNA evidence, other line of evidence that we may use to support the hypothesis. So remember now, I give you this table previous. So that is the evidence for out of Africa, and the evidence for human evolution, and the evidence for evolution in natural evolution or biological evolution. So a lot of questions that they can do. The line of evolution, we start with Ardipithecus, and then the Australopithecus, Homo habilis, Homo erectus, Homo nederlandus, and then the Homo sapiens. That is the line of evidence. Ardipithecus, Tim White discovered Ardipithecus fossil. It was the first one. Ardipithecus ramidicus, it was the first one, and Tim White discovered it. And then we have the Australopithecus, Donald Johnson, Australopithecus africanus, the Taung baby, Taung child. There I made the mistake by saying Taung baby. The Taung child was discovered by Raymond Dart, and that is Australopithecus africanus. Africanus means from Africa only. And we have Australopithecus sediba, namely Karabo, and Karabo was discovered by Lee Berger and his son. Homo habilis, Louise and Mary Leakey did discover them, and Homo sapiens was discovered with Tim White. So will be a short type of question. Who discovered it? ABC question. Cannot count many marks. So make sure I know that favorable is the Tim White that discovered Homo sapiens. Also Lee Berger that discovered Australopithecus sediba, Karabo, which is more recently. Those are the most. And Taung child that was discovered by Raymond Dart. And that is the famous one that they always answer, but that is actually the Australopithecus and the people that discover them that you have to identify on that one. So again, on evolution or human evolution is then the fossils. You must know that we are all of the Hominidae family. Differences and characteristics that we share are very important, and when we explain the differences, we must also look at the significance of that differences. A phylogenetic tree is very important, and then as I stated, please, the learners must go and memorize the out of Africa hypothesis and DNA, mitochondrial DNA as evidence of evolution. They don't have to understand it. It sounds a bit harsh, but they have to learn it out of the exam guidelines. They have to memorize it. They can rap it to one another. They can do anything, but they must read and learn it by heart and by word so that they can be able to give it back in when the question is asked. And definitely out of Africa and mitochondrial DNA is always evaluated in an exam paper, and that is where the most learners loses marks, especially also the level 7 learners. And then on speciation and natural selection, learners must go and read it. and they must apply it according to the exam guidelines, and that exam guidelines must they apply to the example and in that sequence and in the wording that they are giving it exactly as it is stated. So actually on evolution, you can set up a test and just ask the test straight from the exam guidelines. Let them write down natural selection, even if it is the general state, and so that they can get used to the sequence that they must answer and different points that is very important in natural selection and artificial selection. I think it was a long session, but really I can assure you, if you go and look at these key points and you emphasize these key points, especially to level 1 or all the levels learners, there's a lot of technical points and detail that also the level 7 learners must do. But if they focus on these, then they are sure of passing life science. You cannot step into exam room without doing replication, transcription, translation, without not going and look on how to do natural selection and speciation and out of Africa hypothesis and mitochondrial DNA. Those are, if we count the allocation of marks together, it will give you very quickly a mark out of 50. That is more than enough to pass life science. Level 7 learners, go and look how we write the out of Africa hypothesis. Go and look how you must answer questions on natural selection and speciation. And I hope really that you can do this and go and study. And yes, all the best of luck for Friday and coming next Tuesday on paper 2 especially. A lot of content, not so lot of work to learn on question paper 2, but a lot of understanding. So go to the past papers, practice, and best of luck to all of you. I hope this session did help somewhat in some way. Thank you very much.